Hi everyone, my name is Maris and I'm going to be talking about my deep learning final project for this semester, decoding cancer types with microbiome data. So cancer and the microbiome are very interesting in terms of how they're interrelated with each other. And there's, emerging evidence, newer publications that are linking the microbiome to basically the progression of cancer. And so what we know about the microbiome, especially in tumor sites, is that there is sort of a microenvironment that's created. The tumor cells are interacting with the healthy host cells, they're interacting with the immune system, but they're also interacting with the microbiome and getting an understanding of the microbial patterns that emerge when you have, for example, a tumor, a cancerous disease. Then this can really help us understand more of what's going on at the actual cancer site and potentially learn things that can help us to treat patients in the future. So as someone who works with microbiome data, specifically sequencing data quite frequently, my typical workflow for cleaning the data occurs in R. And this is just because of the ease of the manipulation of data sets in R, the ease of visualization. It's a double click on the variable name and an enter, and then you can see everything in it. So my approach was to first clean the data in R and then save it as a CSV, import that CSV in Python, and then from there train my deep learning models. And so the way I approach this, again, similar to how I, approach this in real life in my day-to-day job, is by verifying the sequencing data first. And this is important because if you have like bacteria or viruses that have very, very, very few numbers of reads, it can really mess with your data, especially if you're downsampling at any step of the process, because then you can end up with a ton of zeros and things that just generally don't make sense. So to avoid this detection limit issue, I only take things with above 4,000 reads, and everything with under 4,000 reads per genus is eliminated, not considered in our data set. Then I calculate the relative abundances, and I applied a log transformation, and I centered and scaled this data. And this is because I'm going to put it into a neural network and eventually an autoencoder. And I want to make sure that all the values I'm putting in are within a certain range so that there's nothing that's really skewing the weights for any part of these models and, going to give me NAN somewhere. It all needs to be compact, it all needs to make sense in terms of the actual raw values. So in order to classify different cancer types based on the sequencing data, I started with a feed-forward neural network. And the model architecture was pretty straightforward. I had my input layer, I had 3 fully connected hidden layers with ReLU activation, and I had, at first, no dropout layers, just an output layer after my hidden layers. And this model had very good accuracy, 85%. That's great. But when you look at the loss per epoch graph, as you can see on this top right, it's not that great. You have a validation loss that's trending in the opposite direction of the training loss as the number of epochs increases. This is pretty standard for a model that's overfitting, and that's not something we want because we want to be able to generalize to new data. And so with that in mind, I added some dropout layers into my second feed-forward neural network. And with the dropout layers, you can see that there's a significant improvement in the overfitting issue. The accuracy per epoch suffers a little bit, ending around 82% instead of around 85%. But the loss per epoch, the trend between the training and the validation losses per epoch is actually much better. The gap between them is much narrower, and even though they still do trend in opposite directions after about 30 epochs, it's still a significant improvement over the first feed-forward neural network that I tried that didn't have the dropout layers in it. And so overall, I was getting a pretty good model. I was getting something that was able to classify different cancers from microbiome data with 82 to 87% accuracy. So this was a good start. Even though I had a neural network at this point that gave me a pretty good model, something that was pretty accurately able to classify different cancer types based on the sequencing data, I wanted to try a different approach. One, to try to address the remaining overfitting that I was seeing after I added the dropout layers, but two, to also consider this from a microbiome scientist's standpoint. So in the field of microbiome, a very common approach when analyzing sequencing data is to reduce dimensionality using a PCA or a PCOA. And this is based on a linear approach, whereas autoencoders will reduce the dimensionality in the encoding, but they can handle nonlinear relationships, which was very interesting to me. I wanted to see how it would perform on a microbiome sequencing data set. And so I built an autoencoder, and it wasn't too complex. You can see the encoding layers. There's a dense layer with 128 neurons, a dense layer with 64 neurons, and then it's just reversed for the decoding. And the accuracy was about 83%, which is very close to what the neural network accuracy was when I added the dropout layers. But as you can see, the overfitting issue is mitigated to a greater extent than it was in the neural network models that I previously talked about. The model loss graph shows that the training loss and the validation loss are not really trending in opposite directions, at least not as noticeably as they were before. And again, the model accuracy is still matching that of the neural network model with the dropout layers. Now that I had an autoencoder that could accurately classify different cancer types based on the microbiome sequencing data, I wanted to take a look at what was going on by visualizing the latent space. And that way I could compare it a bit more directly to a PCA, which is what we typically do. You can see that the autoencoder latent space plotted across 2 dimensions shows very clear clustering of different disease types. So each of those different colors is one out of the 10 different types of cancer that I was classifying. And you can see there's a significant degree of separation. You have clusters that are completely standing out from other ones, or that share a well-defined edge with others. And relatively speaking, there's not that much overlap between most of these clusters. So this is a good sign to me. This shows me that the autoencoder is doing a really good job of finding underlying features in the microbiome dataset that are informing it of what cancer type is related to that particular microbiome profile. So this is essentially what we want to see. When you compare it to a center log ratio PCA, as you can see on the right, the PCA has much less refined clustering. Those are the first two principal components, so they'll be the ones that contribute most to the PCA itself. And even across PC1 and 2, you can see the groups are sort of blended together. And there are some diseases that seem to overlap with each other a little bit or stand out a bit, but when you compare it to what we see in the autoencoders latent space, it's not as good clustering. So what's the interpretation of this? We're observing much better, much clearer, more defined clustering between the cancer types in the autoencoder latent space than we are in the PCA. Well, it could suggest that the autoencoder is able to capture very important nonlinear relationships, patterns in the microbiome dataset that the PCA can't capture because the PCA is linear based. And so if this is really what's driving this difference, then capturing those nonlinear features can be a very powerful tool, potentially, in helping us to understand what's going on in this microenvironment where we have the cancer cells interacting with the healthy host cells interacting with the microbiome. So we could be getting a specific microbiome profile to specific types of cancer, which means that all these microenvironments are interacting in different ways with different diseases, and using a PCA, very traditional approach to the sequencing data, simply cannot elucidate that as well as using something more powerful like an autoencoder. As a final test of my autoencoder's ability to really understand these key features underlying the microbiome dataset as it relates to cancer classification, I tested it by getting it to perform a data imputation. I took a dataset the autoencoder hadn't previously seen, and I removed 10% of the data in it. And I had the autoencoder impute that missing data. As you can see in the 2D visualization of the latent space of this dataset with the imputation, the clustering isn't quite as clear as it is in the training dataset, which is expected, but you still see these distinct clusters coming out that, again, you weren't seeing in the PCA. And this shows me that there really is something going on that the autoencoder is capturing, some underlying features in the sequencing data that relate to the different cancer types. So this is further confirmation that autoencoders could be a really powerful tool in understanding the relationship between the microbiome and the host side of things when it comes to cancer. So as a quick overview of what's been discussed so far, for microbiome sequencing data, as it relates to different cancer types, I first looked at feed-forward neural networks and tried to get them to predict different cancer types based on the microbiome sequencing data. And this worked pretty well, got pretty high accuracies, but I saw some overfitting problems, mitigated them to some extent with the addition of dropout layers. But then I saw better performance from the autoencoders in the sense that they had very similar accuracy while having less overfitting issues. Furthermore, I compared them to the traditional approach in microbiome analysis of sequencing data and saw that they outperformed traditional PCA in differentiating cancer types. So this indicates to me that they are better at extracting features from the dataset, and I hypothesized that this is because the autoencoders can capture nonlinear features within the dataset, while the PCA cannot necessarily do that to the extent that the autoencoder can. In terms of future work, I'm interested in investigating the biological relevance of some of these key features that are being captured by the autoencoder but not being captured by the PCA, because this brings us a step closer to clinical relevance. I'm also interested in obtaining more data about survival time, because if the autoencoder is able to predict cancer type based on the microbiome profile with the accuracy that it's demonstrated here, which is very good, then perhaps it might also be able to predict survival time based on the microbiome environment of a cancer site. And so this would also have great clinical relevance. Thank you for watching this video, and I hope you have a great rest of your day.